Now I would like to introduce our keynote speaker for this session, Dina Machuve from Nelson Mandela African Institutions of Science and Technology. She's in Arusha, Tanzania right now, and so she will be taking a Q&A live from us, and we will have her presentation pre-recorded, but you can start interacting in the chat. Her research focuses on developing data-driven solutions in agriculture. For her PhD dissertation, she investigated information logistics of small and medium-sized food processors, and currently she's looking to developing poultry diseases diagnostics tools using deep learning for small and medium-scale poultry farmers in Tanzania. She will be discussing this today. She's an early career fellow of the Organization of Women in Science for a Developing World. She serves in the organizing committee of Data Science Africa, an organization that runs an annual data science and machine learning summer school and workshop in Africa. She's also a member of a research group of Mechanism Design for Social Good. She completed her PhD in Information Communication Science and Engineering at Nelson Mandela African Institution of Science and Technology in 2016. Previously, she was at the University of Tennessee, where she got an MS in electrical engineering and a BS also in electrical engineering. So I'm really thrilled that Dina accepted to present today her work. Gustavo Stolowitzki met her virtually, I think, and again, we're meeting her virtually this year at NeurIPS last year in Vancouver, and was really excited about her presentation. I think Dina, from what I just said, really shows and pushes the values that we have in DREAM. And in general, for the keynotes, we like to explore new continents and new topics from persons that show interest in the open science and data sharing. And Dina is a great representative of that. Being in Tanzania, being a woman, and of course being brilliantly pushing all these frontier ideas in Tanzania. So it's a pleasure for me to introduce Dina, and we can now move to her keynote, and please stay for the Q&A.
Good morning. I'm highly honored to be invited to give a talk at the Dream Challenges Conference that's held in conjunction with the Regulatory and Systems Genomics Conference. My talk today is on poultry diseases diagnostics using deep learning. I'm Dina Machuve, a lecturer in Information and Communication Sciences and Engineering at the Nelson Mandela African Institution of Science and Technology in Tanzania. I'm also a member of the board of Data Science Africa, an annual summer school and workshop that is held in Africa to build capacity in data science. The agenda for my talk today is presenting to you the poultry sector in Tanzania, and also the challenges and the possibilities of using convolutional neural networks in developing countries to address real life problems. And we'll zero into particular to understand how CNNs can be applied for poultry health monitoring. I would like you to guess which chicken is sick. You keep the answer in your mind, and we'll revisit the images later. Tanzania in East Africa ranks third in livestock numbers. However, the country experiences low poultry productivity, mainly due to diseases, and there's hardly data about the sector, although we have chickens population of up to 36 million. And it's a highly economically significant sector, because over 4.6 million households depend on chickens. and our country's population is 56.32. Therefore, addressing the challenges in this sector will contribute to the second goal of the SDGs to reduce hunger and enhance food security. Now, the challenge in this sector is that farmers hardly keep records about managing their chicken health. They mainly rely on the word of mouth. And the chicken diseases are really devastating due to the fact that they bring about high mortality rates, low production rates in terms of meat and eggs. And some of these diseases affect humans. There is a very low number of field officers to assist farmers on these challenges. The monitoring of these three poultry diseases, namely Salmonella, Newcastle disease, and coccidiosis, is mainly done in the lab using PCR from chicken droppings. And it takes about 3 up to 4 days to get the results. And sometimes the diagnosis is done from clinical signs only, and access to these services is quite expensive and limited to farmers. And extension officers who do go to the farm do not have tools for rapid diagnostics of these diseases. We believe that with better tools, farmers will be able to diagnose these diseases on their own. And also with good tools, the diagnosis will be reduced from 4 days to up to 3 or 5 minutes. And once diagnosis is done by the farmer, they'll be able to take the appropriate actions, such as isolating the sick chickens and to reduce, eventually reduce the loss of poultry from these diseases in a timely manner. Um, in Africa, we really it's proven that image data is the better source of data. This is due to the fact that the ubiquity of mobile phones in developing countries is high. In Tanzania, where we have a population of 56 million people, over 48.86 million people, we have over 48.9 million subscriptions. And in Africa, in the continent, we have over 250 million people with smartphones. So really mobile phones present suitable sensor for addressing the different developing social challenges. And also, image data is more suitable as a data format, since we have low levels of literacy and multiple languages. And socially, people really are obsessed with images. And that's what brings us to convolutional neural networks being a suitable solution to address these challenges on poultry diseases. And we understand that convolutional neural networks are specialized kind of deep neural network, which is mainly used for computer vision tasks. And it is proven to be successful in many practical applications, including diseases diagnostics. And the good thing about the CNN is that it's an end-to-end pipeline, where when you have an input image, here in our case is a chicken, you on the output layer, fully connected layer, you'll be able to determine whether it has disease or not. Here in the neural net, we have different representations of the input image at different layers. Different abstractions of the input layer can definitely be abstracted and have different representations of whether the chicken is sick or not. Now, how do we apply this in the field? Here we revisit our images again to see to solve a task whether we can train a model and correctly classify a set of images for to determine the kind of chicken disease that there is. We aim that after training our model, we should be able to return the correct label for each image in terms of even the kind of disease that the poultry has. So this is our end-to-end pipeline where we generate the data set and pre-process the data, and our data set is actually uh, fecal images, and the annotation is done using a vet officer, a veterinarian, or field officers, and we are able to generate the data set, and eventually we'll do modeling, training the images for the different diseases, and we aim to be able to deploy that for farmers to use on the farm. And our model will be deployed on eventually on mobile phone. Now, in Africa, before you apply machine learning, there's a very labor intensive task of building the data set. So we physically go to these farms and take photos of the fecal samples using mobile phones, okay, to take photos of the chicken droppings and take also secondary data of the farms. So we in this task we have to find supportive chicken farmers and build trust with them, and this is where the agricultural experts are very useful, and we normally are accompanied by the livestock officers or vets to these farms, and it takes up to 3 hours to get to a farm and up to 4 hours to collect 150 images. It's really a labor intensive task. And our data set is chicken droppings. And the reason, motivation for using this kind of data set is that clinical signs are similar, and the color and shape of droppings supplement the clinical signs. And this is a visualization of how the different samples look like. This is a healthy, it's quite impact, and this is a dropping for chicken that has coccidiosis. It's quite brown in color, and for salmonella, it's whitish and a bit loose with white color dominating. And so far, we have a data set of 4,924 chicken droppings images, and our target is to reach 8,000 images with 2,000 images per class. There's another disease of Newcastle that we haven't started to work on, and the annotation is done mainly using the vets, but we also are going to use PCR, polymerase chain reaction, to label 2,000 images, 500 images per class. This will definitely be our high quality labeled data set. And the PCR process involves DNA extraction, quantification on the gel, and then doing amplifications with different primers to confirm the presence of the different um, pathogens. And we have started the PCR work initially for Salmonella, and the results are indicated here. Here we only had 3 that were negative, but in this run we had 35 35 samples that confirmed presence of the Salmonella. So in total, when we finish the work, out of the 8,000, we'll have 2,000 that will be high quality data labeled using PCR. Now, our exploratory data analysis on the images includes other features such as the number of chickens that that the farmer has, and the kind of feed that they give to their chickens. And we also have the geocodes. Every image is geocoded, and this is going to be useful on further analysis on prediction of areas with diseases and possible outbreaks. However, we really lack, this has been a challenge. As I said earlier, farmers don't keep um, historical records to track their for management of their chickens. So there's lack of historical data, and our takeaway from the task of data collection so far is that record keeping is quite fundamental for improving productivity of the chicken farmers. And then on the next stage, we aim to train the model. We have so far done baseline training for two classes, the coccidiosis and healthy class, and as a proof of concept. But eventually for deployment, we'll definitely use transfer learning in order to select the appropriate model for deployment on mobile phones. And the metrics that we have used on the baseline is accuracy. So we really see computer vision being a very good solution for pattern recognition, and we've done the work on image classification, but eventually we aim to do as well as localization localization on the chicken droppings. So, you said earlier that after we've done the training, we really see our model should be able to predict the kind of diseases for each chicken. And actually, the first one was healthy. The second chicken is sick but suffering from coccidiosis, and the third one is suffering from salmonella. Now, it's only looking at these clinical signs is not sufficient enough to conclude on the kind of disease that they have, because these two are diseased. But from the chicken droppings, we are able to appropriately say that it has coccidiosis and this other one has salmonella. And our last eventually we really on production we aim to deploy the model on Android, and it will be a free mobile application, and we've selected Android because that's the dominant operating system in developing countries like Tanzania. And we will definitely train farmers and livestock officers to use the app, and we'll definitely serve the model on the mobile app to allow usage offline, and we'll definitely be updating the model regularly online. And eventually our big goal is to have a mobile application for use by farmers and livestock officers for early detection of poultry diseases. And we'll definitely be able to share with the wider research community a machine learning data set for poultry diseases diagnostics. And we definitely believe that farmers who will use our mobile app, this app, will definitely have reduced outbreaks of diseases at their farms. So our progress is here. We're still generating the training and test data set and the high quality data set um, using PCR. But initial results of the baseline model for proof of concept really shows promising results. And I'd really like to acknowledge collaborators and funders, the Organization for Women in Science for the Developing Countries, who is for funding this project, and also the AI for Development Innovation Grant from the Deep Planning Indaba. I also have collaborators, the Delta Analytics Teaching Fellowship, on machine learning, and also the mechanism designed for social good development group. So we are really working hard to reach our target of 8,000 images, and to share this data set with the machine learning community, hopefully by mid next year. And that's my contact and the project website. I thank you very much, and I'm really available to answer any questions at this point. Thank you.
Thanks, Dina, for the great talk. I think we're having some technical difficulties because Dina is in Tanzania. And, as you know, if you grew up in one of the developing countries, electricity goes and comes. So we're going to wait a few seconds to see if she can connect back. But if not, we'll be asking questions in the chat, and we'll try to also respond to the questions on the chat.
I was able to unmute now. Yeah. Pablo, I'm here.
So we already have a couple of questions from Irene Kapolov from Carnegie Mellon University. We're trying to just see if Dina can come online and answer them speaking, or if we'll just move to the chat and try to answer the questions in the chat. I think, I think her talk really exemplifies the type of applications that image classification can have in contexts that we are not aware of. So it is known that mobile phones, and the fact that all mobile phones have cameras, are a power for collecting data. And Dina really has shown how you can push this. Still, as in many of our dream challenges, the problem is actually collecting the data, and you've seen the time it takes to actually physically go to these places to collect the initial data. But I think once you have a sufficient number of images to train your model, then as she showed, the model can be applied directly by just putting it in an app and allowing users to do the image classification themselves. So in this case, the application was trying to classify chicken droppings into whether they were disease or not, Salmonella and coccidiosis. Which is an important application in Tanzania, I guess, in these remote places where you don't have a veterinarian to help you diagnose your chickens. Okay, so Dina will be responding on the chat, and I guess I will be passing along the responses. It's, as she says, it's raining in Tanzania, and so the connection is complicated. And also, a couple of weeks ago, there was an election with some turmoil. So, the field conditions in our countries in general, or the country I grew in, are not as stable as, in first world countries. So it's always amazing to see how far people can go to, continue doing science and education. And I think this is part of the passion that we have also in Dream. So I'll just read, here's Dina responding the questions on the chat. I guess I'll just also read them in case there are not people in the chat. So there's a question from Irina Kaplo, which is, Are you training your model on other image classification problems? And if so, how are you doing the pre-training? Dina responds, we are pre-training the model on other image classification problems. We are creating a high quality data set that will be labeled using PCR. Another question from Irene Capua, which is, you mentioned that you plan to update the model online. When you update the model, will you retrain the model from scratch or initialize the model with the existing model weights and then fine-tune those weights with the new data? Dina's response is, this high-quality data set will have 2,000 images, 500 per class, and we'll pre-train, and she will pre-train it with another data set with 8,000 images, 2,000 per class. So still a lot of work to do on the field. And Dina also says that she will fine-tune the model with new data on updating the model. I guess I'll also ask a question and I'll write it down. So Dina, how many people are involved in collecting the data for the model? So with a team of 9 people, Dina is able to collect all this data in the field. And did you initially try to use pre-trained models that you could? She's saying that she on average she collects send 100 images a day. Did you initially try to use pre-trained models on image recognition, or that you could adapt, or you have to go back from scratch and building on your CNN model? I see. So she has used VGG16, and so far the results are promising. And so, if I understand correctly, is the idea that farmers will send the pictures, or will they download and run locally? Okay, so the farmers will download the app and use it locally. Cool. So Gustavo Starovitsky has also another question, which is, the image you show that difference in the color, and it seems simple. Is the task in reality much more difficult? Can you estimate the degree to which a chicken is sick? So the question, I guess, is around the fact that how much more does analyzing this with a CNN brings to the knowledge? Are there things that are not so obvious initially? Right. And so I guess I have another. So Dina just answers that the clinical signs are normally not obvious. Another question I have is, what percentage of the images, the chicken images you've taken, are show disease by PCR? What's the prevalence? of the disease. Can we try again if her sound works, maybe? Or okay. So she said that so far they have results on PCR for only one disease. Was this salmonellosis? And what is the prevalence in the samples you've taken? What is the prevalence? Because I think you said you have collected already. How many images have you collected? Sorry, I missed that part. Just to know how prevalent the salmonella is in the samples you've taken. Oh, 70% prevalent. Wow, that's really a lot. So it's a really prevalent disease. And I guess that, yeah. So are you planning, what would be other types of industries where you could apply this type of approach in Tanzania or in other African countries? Would this work with I don't know, cattle or agriculture? I mean, do you have ideas of other applications for agriculture? Right. So she's trying to the hope is that the app will be used in East Africa and Asia. Very good. I mean, it's a bit sad that we couldn't discuss. I mean, I had a lot of questions about, how you decide to go back to Africa and, the types of, the app will be free. The types of how you're trying to push, the use of artificial intelligence, and also the hackathons that you're organizing, and how you're trying to push these types of questions in Tanzania, and I guess through your students in the university. I think it's a little bit hard to discuss this just in a chat. It would be better if we could discuss it live. I think there's another question from Justin, our chair, if you would be interested in hosting this study as a dream challenge to see if we could get even better algorithms for doing this type of classification. And I think, it will also, as I say, will be also interesting to see if we could apply it to other types of disease in other, in cattle or in other types of animals. Okay, so I think it's great. So Dina said that she would love to host a dream challenge. It would be great to do this. We're getting interested a lot, of course, in image classification and the application of convolutional neural networks to everything that can be basically encoded into an image.
Sorry, is it still noisy? Sorry.
Yeah, hi. I think it's still a bit noisy. There's a strong, I guess, background that comes in and out. No, it's okay. I mean, we knew this was something that could possibly happen. I think are you still there? Can you hear me? Oh no, you muted yourself. Okay. Yeah, we knew this is something. Yeah, this is something we knew could happen, but we really wanted that you present us the type of frontier breaking things you're doing and all the work you're doing to push these beyond just more academic applications and all the field work that you need to do for this. So I mean, it's really a pleasure to have you here and then you presented the type of things you're doing. Really inspiring, and I thank you a lot for this. I think we will move on to the next session. We're a little bit early, but it's sometimes good to be early. So thanks a lot, Dina. So I want to just pass now the torch to Brian White from Sage Bionetworks, and he's going to be presenting us the Beat AML challenge. Brian.